Good evening and welcome to the October meeting of the Pinal Gem and Mineral Society and the Pinal Geology Mineral Museum from Coolidge, Arizona. Tonight we have Dr. David Gillette, and he will join us. He has joined us, and we will talk about Glyptodonts, the most famous Ice Age mammal you've never heard of. Dr. Gillette, until recently, was the Edwin Colbert Curator of Paleontology at the Museum of Northern Arizona. He recently retired from that, and so he has time to do our talk tonight. I'm going to give over to him. He will be able to talk with you, introduce himself a little bit better than that. I'm excited because I'm a dinosaur fanatic and he discovered Seismosaurus, so I'm really excited to meet him. But I'm going to turn it over to him and we'll get started as quickly as possible. Okay.
Thanks, Bob. Can you hear me? Can anybody hear me?
Yes.
Okay. Thanks for the plug on Seismosaurus. I should introduce myself a little bit. I'm probably more well known for work I've done on dinosaurs over the past 30 years, but I also do a lot of work on mammals and started my career on mammals, and that included Glyptodonts that I'm going to talk about tonight. I retired from the Museum of Northern Arizona after 22 years in July, and immediately became a volunteer, and it's just like working. The only difference is I don't get paid and I don't have to go to meetings. So there's a trade-off. Before that, I was state paleontologist of Utah for 11 years, and then I had several university positions before that. And I've been a paleontologist since, I reckon, around 1968. So I've been around for a long time. I'd like to tell you about one of my favorite subjects, or talk about it today, and that's Glyptodonts from North America. My study began 48 years ago at the Smithsonian Institution, and I had a fellowship to study animals that look like this. The upper picture is a restoration based on the skeletal anatomy shown below. And this is an adult Glyptotherium texanum. It used to be called Glyptotherium arizonae, but it turns out texanum and arizonae are the same species, and so texanum was named first, and it had priority, unfortunately for us in Arizona. The signature part of that skeleton is the armor, which consists of around 2,000 bony plates that in adults were fused to a rigid condition, except for in the front of the body where there was a little flexibility around the shoulders. And those plates are called Osteoderms, and they're typically hexagonal or pentagonal. And as thick as 2 inches, and they fuse together to form a shell, a lot like the shell of a turtle functionally, but turtles only have about a dozen plates that fuse during development, whereas Glyptodonts have 2,000 of these bony plates, or maybe even more. And the tail was also covered with bony plates. These are those bony plates. These are called osteoderms, sometimes also called scutes. And these are about 1.5 inches wide and about 3/4 of an inch thick. And they firmly attach to adjoining osteoderms on their straight edges. And the typical anatomy is 6-sided, but it's not a perfect pattern of 6-sided osteoderms through the whole body, because the body shape changes and the osteoderms changed accordingly. So these are thick, heavy bone, and those pits that you see, especially the big ones that are in those grooves, allowed for the growth of hair and bristles that were sensory, so that if an oxpecker were to land on the shell, they would know that there was something on their shell. So that there was a function for those holes, and the smaller pits were nourishing, had blood vessels that nourished the skin and scales that covered these individual osteoderms. So this is Glyptotherium and Glyptodon. Glyptotherium is the one in North America. Glyptodon is a close relative in South America, but there were around 40 genera of Glyptodons in South America. Only one made it to North America, and that's the one we call Glyptotherium. So my work started at the Smithsonian in Washington, D.C., at the Museum of Natural History. You can see outside and inside here. And it was an exciting place to work in the 70s. And I was allowed to work on Ice Age animals on my fellowship. And I walked by this giant elephant every day on my way into work, or in my way into study is a better way to express it, because I was doing research. And this was the largest known elephant ever taken. So it was an exciting time every day, and I never got tired of it. So here's that elephant. It was a magnificent mount, and it greeted visitors in the rotunda, people from all over the world. Here are two pictures of collections at the Smithsonian. The paleontology collections are organized about the same way, but I couldn't find any pictures of them, so I took these offline. But this is the nature of the collections at the Smithsonian. And one of the really wonderful things is all of those collections for fellows like I was were available, both the biology and the geology collection. So it was a wonderful year. I actually extended that to about 2 years, from 1972 to 1974. And my work was centered on relatives of the ground sloth, the giant ground sloths shown here on the left, that are also relatives of armadillos. And you can see the carapace and shell of a Glyptodont underneath this giant ground sloth. So the Glyptodonts were nowhere near as large as the ground sloths, but they came into North America at the same time from South America, and they're actually rather close relatives. They belong in the order called the Edentata. The Glyptodont, the word Glyptodont means grooved tooth. That was a name coined by Richard Owen in 1839 when he received a partial skeleton of a Glyptodont from South America. And you can see that he got a little mixed up in some details of the anatomy and didn't really know how those osteoderms fit. But you can see the occlusal surface of the tooth in the upper right, and a side view of a tooth in the lower right. And those grooves in the teeth is where the feature that Owen chose to coin the word Glyptodon, or the name Glyptodon for the South American genus. So that name means grooved tooth, and all Glyptodonts have teeth like the one shown here. But a really odd peculiarity in Glyptodont teeth is they have no enamel. Those are naked teeth, so to speak, and you're looking at different variations of dentine in the anatomy of those teeth. So here's a skeleton of Glyptodon on the left and a restoration by an artist of Glyptodon on the right. They're not like any other animal. There's just nothing that resembles a Glyptodon except for armadillos, and armadillos are tiny by comparison. The skeleton on the left is a display specimen in South America. One of the Glyptodonts that is famous and often included with toy dinosaurs, as though it were a dinosaur, is the one called Dedicarus. This is the one with the bony mace on the tail that it could use for protection. That long tube leading to the mace on the tip of the tail is solid bone, and the rings near the body for the tail also protected the tail. And this was a pretty mean dude in life. I think they wouldn't have put up with much nonsense, and they could very easily protect themselves. Though Glyptodon on the right is a Glyptodon for comparison. They didn't have a mace on the tail, but they had bony spikes on those rings on the tail. So this is Dedicarus on the right with people in the picture, and Dedicarus is nearly as big as a Volkswagen Beetle. There's a model in the lower left of the picture here showing how Glyptodon resembles a Volkswagen Beetle, but I think an even better comparison is Dedicarus. And Dedicarus was as large as a Volkswagen Beetle. So this is an animal that probably weighed about 2 tons. Half of that weight was in the shell with all those bony plates. Here's a skeleton of Dedicarus in a South American museum. And it's one of the weirdest animals you could ever experience. So the Glyptodonts I studied at the Smithsonian came from southern Arizona, came from the Safford area. And this is a mounted skeleton in the upper left, and it's the same skeleton looking head on in the lower right. And then a side view while these were on storage in the Smithsonian as they were revising their current displays. If you look head on at the Glyptodont, you see 2 weird holes in the skull on the sides. Those were holes for muscles for the jaws that attach from the back of the skull and on the side of the face. And so those are not the eyes, those are openings for muscles of the lower jaw. And they probably fed all the time and spent all their daytime hours either in combat or feeding. Here's a BBC reconstruction of Glyptodonts that was done after BBC participated in the 2012 excavation in Safford. And they got so excited they did computer-generated animation for these Glyptodonts, and I was a consultant for these drawings and the behavior. It was in a documentary called Ice Age Giants, and we'll talk about that shortly. So these are their sketches of Glyptodonts. This is Glyptotherium texanum on the lower left, and then juvenile on the right-hand side. And that's that shell in the excavations was upside down, and we're going to talk about that shell, and you'll see that drawing in a moment. Here's another view of Glyptodon in a museum in South America. I took this photo and then looked at it later, and I had nightmares over this thing. This is so scary. And here's Glyptodon on the right with 2 different kinds of ground sloths in a museum in Argentina. Ground sloths and Glyptodonts are common in the Ice Age in Argentina, and the variety is just astonishing. The only really important predator on Glyptodonts and ground sloths and the other animals that lived in the Ice Age before the exchange between North America and South America was this terror bird. The terror birds were giant predators. I think they were something like 6 or 7 feet tall, and they could rip apart their prey without any trouble at all. They were fast, bipedal, and basically a dinosaur on 2 legs with feathers. And that's probably why Glyptodonts had their armor as protection from the terror birds. Later, predators from North America got into South America, predators like saber-toothed cats, and that armor served the Glyptodonts just as effectively as when it was used for protection from the terror birds. Here's another diorama. And here's a a little bit of action figure activity, the Dedicarus in combat, either competing or in combat for territory. Nice drawings here, and a lot of speculation about how powerful that mace was. Here's the skull of Glyptodon. It's very much like the skull of Glyptotherium. My restorations of Glyptotherium are shown in the bottom. Glyptodon and Glyptotherium are sister taxa, and we use the well-preserved skulls of Glyptodon for studies of functional anatomy of the skull. Before we get into excavation, I want to talk a little bit about stratigraphy. Glyptotherium texanum in Mexico showed up around 3.6 million years ago in central Mexico. And that same population expanded slowly into North America and showed up about 2 million years ago in the Safford area. We're dealing with the same species early in Mexico, and then that population expanded into southern United States all the way to Florida by the end of the Pleistocene, the end of the Ice Age. But in Safford, the age is about 2 million years. So we did 2 excavations that were really exciting in Safford. The arrow shows the location of those excavations. The beds were from the Pliocene, late Pliocene, early Pleistocene, dating from about 2 million years ago, and they had associated fauna including mastodons, saber-toothed cats, extinct horses, ground sloths, camels, tapirs, lots of reptiles and turtles. And it was on property managed by the Bureau of Land Management, and these excavations were done under permit. The repository for all of these collections is the Arizona Museum of Natural History in Mesa. So let's talk about these excavations. This is the Glyptodont carapace, right side up. The head would have come out of the right side and the tail out of the left side. BBC artists drew this for us, and the way they drew it was to show it upside down in this illustration, because that's how both carapaces were excavated, how they were found when we excavated them. They were upside down, and this is a mystery. Why were these shells totally upside down and perfectly positioned as though they were an urn or a vase? I have some ideas about that, and we'll talk about it after we talk about the excavation, but this is how those shells were buried in the sediments. This was in 2010 in Safford when this shell was first discovered, and you can see some bones in the ground. We were beginning a trench, hoping that it was going to be a complete carapace, and it was. This is that same carapace in excavation. We've covered it with, we dug trenches around the sides. We've covered it in plaster and burlap, and we're getting ready to add 2x4s and steel bands to hold it together. And my model in the lower left shows the orientation of the skeleton for each of the photos. So you'll see that a picture of this upside down Glyptodont toy in each of the photos just for orientation. Here we added 2x4s and plaster and burlap. We're undercutting. The next procedure is to undercut that pedestal and cut underneath in the middle to make a tunnel, and then run plaster and burlap upside down in that tunnel to lock everything together, and then gradually reduce those 2 pedestals and add plaster and burlap and lumber to lock everything into place for eventual transport. So that's that carapace upside down in position as we excavated it. That's Larry Thrasher in the center with the hat on. He was the BLM representative and manager for Paleo, and was a great help for all of our work. Here's that carapace completely encased in plaster. It's ready to be lifted out and carried to the lab. It was taken out by a forklift provided by local farmers, and it probably weighed around a ton or a ton and a half. That's with all the rock inside and all the sediment still in place. And then we moved it to the lab, took the top off, still upside down, and began extricating and exposing the bones. And the lower, in the left side, you can see where that centimeter scale is. That's the articulated rear foot. I'll show a picture of that in a moment. And all of the bones in the foot were still in place just as they were buried and just as they were situated when this animal died. So the ligaments and muscles held everything exactly into place, and they got buried and locked into position. And it's a wonderful thing to find a foot that's that beautifully preserved. Here's that same block showing tail rings. Ring number 1, 2, 3, and 4. Those rings each enclosed a single tail vertebra or caudal vertebra, and those rings were solidly fused osteoderms that were curved and locked together. In the outer row of each of those rings had a spiny anatomy for discouraging predators. Beautiful, beautiful preservation, and really a wonderful experience to see this come out of the ground. Here's that articulated foot looking down at the ankle in the top view. You can see the toes all still in alignment. This is a lot like an elephant's foot, which makes sense because it's designed for moving a very heavy-bodied animal. Exciting for paleontologists like me to see bones like this. Then in 2015, we did another excavation that was a carapace found on the side of a hill not very far away from the one in 2012. That's Larry Thrasher pointing at the existence of bones on the side of the hill. You can't see them very well here, but they showed the outline of a carapace upside down. And it's in this picture. I don't have a pointer handy, so I can't point to all those bones, but you can see one V-shaped bone in the upper part of the photograph and some plate-like bones near the notebook case on the lower right. This is the beginning of the excavation. A couple days into the excavation, as we dug a trench around and began exposing the carapace, we had exactly the same situation as in 2012. This carapace is upside down. The plaster loop marks the boundary of the carapace on the outside, and there's bone showing on the inside that soon got covered in plaster and burlap to lock it all in place. Here we're mixing plaster and burlap and applying it to the top to lock everything together. And this allowed for the beginnings of trenching, first vertically and then trenching underneath to create a pedestal. And as we do that, we add more plaster and burlap around the rim and then around the sides. Here it's all in a vertical pedestal. We're adding plaster and burlap. Here we have narrowed the pedestal, and we're adding plaster and burlap underneath. And there we narrow it further. It's a little scary sometimes when you narrow one of these pedestals, because if you go too far, the rock will start to fall out from the carapace. We were dangerously close to the carapace and didn't really know that. We knew the shape of the carapace, and so we could predict what shape we wanted our block to be. But we didn't realize we were only about 2 inches from the carapace itself as we narrowed the pedestal. This was a pretty exciting excavation, and the carapace was in just absolutely beautiful condition, so we found out later. So here we're narrowing the trench, digging underneath. The goal here was to get a tunnel cut in the middle so we could wrap plaster and burlap to lock it all together in the middle, and then narrow the 2 pedestals that resulted after that tunnel was built, and pretty soon have it completely encased in plaster and ready to be moved. Here it's completely encased in plaster and ready for forklift to come move it out of the quarry, provided by the BLM. This is a big job. You can see there's a lot of dirt to move, and it was heavy labor. So here it is, all alone in the quarry. I think I call that a work of art. That's my kind of art. And there it is on the side of the hill to give context. So that was covered by sediment, part of the 111 Ranch sedimentary rocks that occur in the area, now exposed. And here it's being removed by a forklift. And I wasn't there. They said they could handle it. So I said, okay, I had to go back to Flagstaff. Here they're moving it toward a flatbed truck. Here's that flatbed truck. And they turned it upside down. And here's what turned up underneath. That's the Carapace. You can see the pattern of the Osteoderms. The rock was only about 3 inches thick here, and it had fallen apart, and everything stayed exactly into place, just the way we always hope it would. Here's a close-up of that same shot, and you can see how solid the Osteoderms are fused together. There's a crack in the Carapace. That was probably a crack before we ever started excavating. You can see roots and other debris in position there. So we didn't create that crack, and but it doesn't really matter because that's easily repaired anyway. So this was pretty remarkable, and it was exposed at the site before it was transported back to Mesa to the museum. So I'd like to go back and review what I did in my dissertation. This was at that time called Glyptotherium texanum. It's still called Glyptotherium texanum. This one was from Texas, and it's the type specimen of Glyptotherium texanum. It and another skeleton from Safford were the two main skeletons I had to deal with, and I wasn't sure if they were juveniles or adults, and so that became a problem that I wasn't able to solve until 30 years later. This is a relatively small individual, not very different from small individuals that had come out of Safford that I had available at the Smithsonian. At that time, I wasn't able to determine how many species there were with total confidence, but I had reduced the number of species named for North American Glyptodonts to 5 and had reduced the number of named genera to 1 out of what had been 5 or 6 different genera. So I reduced it to 1 genus, Glyptotherium, and then 5 species. The 2 early ones were Glyptotherium arizonae and Glyptotherium texanum. And it wasn't until we got baby and juvenile individuals from central Mexico, just by good luck, that we were able to recognize how those osteoderms grew. All 3 of these pictures are drawn to the same scale. So you can see the increase in size for osteoderms and the changes in the geometry of that pattern of grooves on the surface. This allowed me to figure out that Glyptotherium texanum was a juvenile. The Safford individual that I studied at Smithsonian was a juvenile. But then we've gotten adults out of Safford in the last decade and adults from central Mexico. And I realized that Glyptotherium texanum and arizonae were the same species based on an understanding of the growth. This is why having a good sample is important in paleontology, so we can understand variation due to growth and variation due to geography or just random variation in populations. So North American Glyptodonts have, by my recent recalibration, reclassification, one genus, and it has two in-member species. That is, Glyptotherium as its earliest species, Texanum, and its latest species, Cylindricum. I don't think there's a very good distinction even between those two species. So over a 4 million year period from the earliest Glyptodonts in Mexico all the way to the end of the Pleistocene in North America, I think we have only one species, which is an interesting case of evolutionary stasis. The more conservative interpretation is that we have two species, one in the early phase and one at the later phase at the extinction in North America. And Glyptotherium cylindricum is only slightly different from Glyptotherium texanum. And this is a case of evolution without branching. So it's the same population, the same genetics, the same species, all the way through the Pleistocene, but ending up with a species that is just barely different enough from Texanum that we can conserve the name Cylindricum. So it's a very slow evolutionary story. Here's that story shown in a diagram. Early Pleistocene in central Mexico, we have Glyptotherium texanum. By the late Pleistocene, all across the southern United States into Oklahoma and as far east as South Carolina. By the end of the Pleistocene, it's Glyptotherium cylindricum, but no branching in this evolutionary history. This is a big surprise because we thought we had 5 different species and a complicated story of evolution. Instead, it's a very conservative story in the evolution of these species. So here's a twist on this story. During the middle and late Pliocene, North America and South America became connected at the Isthmus of Panama, and this opened the doors for terrestrial animals to expand their populations in both directions. I don't like to call it a migration because they weren't really migrating, their populations were just expanding. So South American animals, including Glyptodonts and armadillos, possums, and ground sloths, expanded northward, and North American animals like saber-toothed cats and mastodons and mammoths and camels were expanding their population southward. So this was a great biotic exchange that has come to be known as the Great American Biotic Interchange. And it started about 4 million years ago. And I made a little cartoon here showing Glyptodont and Dedicarus. Those are South American genera. They're headed toward North America. Glyptodont says, it's getting crowded here. I'm going to North America. I'm out of here. And when I get there, I'm going to change my name to Glyptotherium. Well, that's a cartoon version, but that's really what happened. Glyptodont populations of the genus Glyptodon expanded into North America, and somewhere along that line changed enough evolutionarily to be a different genus. And we tested that idea recently and confirmed that Glyptodon and Glyptotherium are in fact different, even though they're closely related. Here's the range in the population for Glyptodonts in North America and Central America. And we have to remember that during low stands of the sea, during the Ice Age, the continental shelf offered almost double the amount of habitat that was available during high stands of the sea. And I expect there's a lot of Glyptodont fossil deposits on the continental shelf, along with mastodons and mammoths and other animals of the Ice Age. My lines are a little out of whack here on this diagram, but you get the idea with the continental shelf. So there was lots of area for all these new animals to evolve. Well, here are some photos of another Glyptodont that had come from Safford earlier, and this is on display at the Arizona Museum of Natural History in Mesa. It's identical to the female version of Glyptodonts that we recognize, and the lower left is a view looking straight down from the top. So that's my shirt there, and that lower part of that photograph, and buttons in my shirt. And I reached as high as I could and got a photo that showed the very top and the shape of that carapace. The other two pictures on the top are that same carapace looking from the side. The hole in the right-hand side is the opening for the tail. And you can see that the opening for the tail there doesn't have big spikes. And I think that's a mark of the female version of the carapace. The males have big spikes on that ring of osteoderms that cover the top part of the tail. So these are on display at the Arizona Museum of Natural History. They're very striking. You have to sort of search them out because there are bigger animals around them and dinosaurs nearby, so it's easy to get excited and walk right past them. But these are really spectacular and beautifully preserved. So there's another puzzle that I had to deal with in my dissertation. This skull on the right came from the Safford area. It was excavated in the 1930s by the American Museum of Natural History in New York. They sent out excavation parties during the 1930s and did a lot of work in southern Arizona. This skull is a juvenile, and once it was fully prepared by technicians, we realized that there were two puncture holes in the top of the skull. Those are not eye sockets. The eye sockets are the big circles in the front, but those two puncture holes are the same size, they're elliptical, and they're parallel. And it was a difficult story to put together because in life, Glyptodonts had all these osteoderms on the top of the skull called casque osteoderms. And here's the explanation. There were saber-toothed cats in that fauna, and often cats, including saber-toothed cats, would dispatch their prey with puncture holes right into the brain. So we know how this individual died. Those two holes are from the saber teeth of a saber-toothed cat. We're not sure which genus it was, but it's a dramatic display. This is on display in New York City at the American Museum. And I always love seeing this one. So here are two Glyptodonts in combat. If you want to see computer-generated animation, you can go to the YouTube trailers. The BBC has taken the full show off of YouTube, so you have to buy the series called Ice Age Giants. And Glyptodonts are in the middle part of part one, and that included the excavation in Safford with BBC participants working with us and a lot of action. So if you're so inclined to want to see a little more adventure, you can check out these trailers, and if you can get a hold of a copy of that documentary, I think you'll enjoy seeing that. One big question was, why did Glyptodonts become extinct? Well, in South America, there's actual evidence of humans and Glyptodonts interacting. Around 1900, there was a story in a science journal as a report that people were using the shell of a Glyptodont, probably a Dedicarus rather than this Glyptodont, but a shell of a Glyptodont to live in. The bony plates were so solidly fused that they gave an adequate shelter for people living in South America about 11,000 years ago. There's actually no evidence that they were used for food, no evidence of butcher sites, but I wouldn't doubt that they were easy to take and gave a lot of good food to early people in both Americas. So we don't know really why any of the Ice Age animals became extinct for sure. Probably overhunting was a factor, but also climatic change and rapidly changing habitats due to fluctuating sea levels. And they went extinct along with mastodons and mammoths and saber-toothed cats and camels and horses and all the animals that lived right up to the time that they became extinct at 11,000 years ago. So I'd like to acknowledge some of the folks that have helped us out. The Arizona Museum of Natural History has been wonderful as a collaborators. Bureau of Land Management in Safford, the Arizona Wildlife Museum in Tucson, the National Museum of Mexico, the Museum of Northern Arizona where I worked, and then 2 organizations in Argentina have individuals with whom I've collaborated and written papers about Glyptodonts. And every time we write a paper, we learn a little more. So this is artwork and photographs of Glyptodonts, and hasta luego, amigos. See you later. And I can take questions now.
Okay, well, thank you, Dr. Gillette. I'm waiting for some questions to come in. Let's try that. I'm having a little technical difficulty tonight. All right, we're hoping for some questions to come in from the comments. We haven't seen any yet. We do have visitors from Texas and Ireland and Mexico and Michigan and California.
All right.
Here we go. You mentioned sexual dimorphism in Glyptodonts. What characters can you use to distinguish them?
The characters are pretty subtle, but the females have smooth osteoderms around the caudal opening, where in the shell where the tail emerged. And males have very long spikes, not spikes like on the mace of the tail of Dedicarus, but they were very pointed. And the explanation for that is the smoother contours of the carapace in the females allowed for more comfortable mating. So that females and males had different anatomy, different roles to play, and that's one of the differences. We also see differences in the size. The females are smaller, males are bigger. That's often the case in mammals, but sometimes in mammals, females are bigger and males are smaller. But that difference in size seems to hold along with the difference in the anatomy of the tail. I actually thought that those were different species. The male was Glyptotherium arizonae. That's the way it was originally described in the 20s. And I thought that was a valid species. Glyptotherium texanum had the smooth carapace openings around the tail, and that was Glyptotherium texanum. And so texanum was female, arizonae was the male. Now we know that was all one species.
Okay, we have another question. Why were those fossils found upside down?
All right, good question. It's one we talk about a lot. Larry Thrasher was the first to recognize that most of the Glyptodonts in the Safford area have been found, the shells have been found upside down, and they're perfectly upside down like a cup. I think they weren't found upside down because of the way they died on land, but I think they died in water, belly up, and sank in water into mud, gently into mud, and then more mud and sediment filled in around them that held them up. If they had died on land, they would have fallen over sideways, but never gone upside down. So I think they fell into water, and I think, I haven't been able to document this yet, but I think most of the sediments in the beds that have Glyptodonts in the Safford area are ash beds that have altered to clay that look like clay stone now, but there's no bedding in that clay. And there's a lot of greeny material that I think is residual ash. So I think this might have been a big ash flow that filled in around the bodies before they had time to disintegrate. So they died belly up in water, they floated, they sank, then they got filled in around the sides, maybe with ash that held everything together instead of the skeleton collapsing. That's the best explanation I have
Cool.
at this point.
All right. What are the parameters or characters to delimit the growth stages of the Glyptodonts?
Good question. We didn't have a very good handle on that early on, 45 years ago. Now we do. The babies, which we know from Mexico, have very small osteoderms, and they're very thin. Osteoderms in juveniles grow in diameter but increase greatly in thickness, and in adults they continue to grow in diameter and continue to increase in thickness. So individual osteoderms chart the growth of individuals. I can give you an estimate of the life stage of an individual if you give me an osteoderm of Glyptotherium. The teeth also change during development. The teeth are conical to begin with in the babies. The teeth flatten out from grinding as the teeth grow, and they become prismatic so that all the sides are parallel instead of conical in juveniles, and then more parallel and larger in adults. We also see changes in the skull. We have just enough of a sample of growth stages to be able to chart those differences.
Great. All right, one more. Are there any differences in the osteoderms between Texanum and Cylindricum?
The answer is yes. Glyptotherium texanum has sculpturing, those radial grooves in the surface that have a circle in the middle, and then radiating grooves that go to the perimeter for each osteoderm. That central figure in Glyptotherium texanum is always slightly larger than half the diameter of the osteoderm. In Glyptotherium cylindricum, that central figure is always slightly smaller than half the diameter of the osteoderm. That's a very minor difference between the two species, and that's the only consistent character I can recognize that distinguishes Texanum from 4 million years ago to Cylindricum at 11,000 years ago. And that's why I say there was very little evolution. I'm not even certain that that difference is accurate. I need more specimens. So that's the best I can do on telling the two species apart.
Okay, that seems to be all our questions. I'm going to thank you very, very much. I'm going to also say I also worked at the Smithsonian at Natural History Museum, and I got to work with a lady in the blue suit in your bird picture. So
fantastic. Small world, isn't it?
Yes, it is. All right. Do have one more question that's popped up.
Okay.
Did they have live birth or lay eggs?
Well, they certainly had live birth. They're technically a variation of armadillos genetically. That was a study recently established from fossil genetic material that geneticists were able to extract from Glyptodonts, and it was a very close match to armadillos. So there's no question that they're about their relationship. Armadillos give live birth. The 9-banded armadillo, strangely enough, gives live birth to, I think it's 8 identical same-sex individuals.
Yes, they're all
weird.
all twins.
Yeah, they're basically octuplets or something like that.
Right, and they're very useful. They're the only other mammal that gets leprosy, Hansen's disease, and so they're very useful in Hansen's disease research, especially because of that live birth.
Yeah, exactly. I collected armadillo skeletons for a while from roadkills, and then when I learned they carried leprosy, I decided, well, maybe I have enough skeletons.
Yeah. All right. Well, I'll say good night to you, sir. Thank you very, very much. I just have a little bit of business to take care of before I turn off the cameras.
Okay, thanks, Bob.
Thank you, and I'll send you the link to this and everything else.
Thanks, everybody.
Okay. And you were interested in how many visits we had. Top viewership of 28 at a time.
All right, good turnout.
Morley, bye.
Take care, everybody.
All right, so come back, everybody, in November 18th. At not necessarily the same time, because the time is changing and Arizona does not change time. So we will join the mountain time zone in the U.S. instead of the Pacific time zone. But here in Arizona, it will be 7 o'clock and I'm not sure anymore when it will be California time, but it'll be 7 o'clock Central. We will have Tisha Stewart, who is a National Geographic environmental educator, talking about aquifers. And it should be a very interesting talk. She's an exciting person to listen to, and I really thank you for coming. Thank you for watching. Thank you for your questions. I hope you enjoyed the show. I'm going to turn us off now. Thanks very much.